This is TWIS. This Week in Science, episode number 881, recorded on Wednesday, June 22nd, 2022. What is crawling on your face? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with meat, mites, and pee. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Once upon a time in science, there was a question. It wasn't the first question, nor would it be the last, but this particular question had an odd sort of ring to it. One that would echo throughout time and change the way things once were into the way things are now, and it continues to change the world now and on into the future. It had an odd sort of ring to it because, unlike other questions, this one you could ask about anything. Without changing a word, you could ask it about anything. And the answer to the question, if you followed it through, could change dramatically, depending on when, where, and about what you asked. Once upon a time in science, there was a question. First spoken in a language you have never heard, a simple ordering of words or meaningful grunts, a mere emotive gesture maybe. That conveyance of a post-observational inquiry started humanity down a path that no other creature that has lived upon the earth has been so emboldened as to traverse. Once upon a time in science, there was a question that we here on this show, at the cutting edge of scientific discovery, on the tail end of future modernity, echo each week as it is passed along through scientific study. A question that is now so imbued within the genetic memory of our corporeal existence that it permeates the minds of children still too young to speak it aloud. Foundational to the very essence of our curiosity, the catalytic reaction driving every one of humanity's accomplishments. And still, the most important question we ask today. Once upon a time in science, there was a question. Huh? I wonder why that is. And we will follow the answer to that question across an assemblage of scientific subjects here on This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about politics. No, we're here to talk about science. Of course, we're here to talk about science, and I'm so excited that you're here with us. We have so many great things to talk about today on this World Day of Camels. Yes. It's Camel Day. It's also Rainforest Day. So let's all be.
And yesterday was Giraffe Day.
Yeah, it's yesterday. It doesn't count.
That's yesterday's news.
Yeah.
It's getting a little long in the neck. Anyway, I have stories about eating this week, meat and plants, what your breath says about you, and a bit about an itch. What do you have, Justin?
I've got pea farming, what standing on one leg says about you, ancient English before the Brexit, and cure for cancer that comes from a plant virus.
Ooh, very exciting. Okay, I'm interested in all these stories. Blair, what's in the animal corner?
I'm going to have to wait and figure out if that was PEA or PEE, but we will find out.
We will learn.
I brought what everyone wanted to hear about this week, face mites. And then, of course, in the animal corner, I have frogs that are bad at jumping and bats that are good at remembering that also eat frogs. So
Bat frog, bat frog, bat frog.
yeah.
Frog bat. It's a new superhero. I'm Sure of it. Transitions back and forth. As we jump into the show here tonight, I would like to remind everyone out there that if you haven't yet subscribed to TWIS, you can find us places that podcasts are found. Those places are many and various, but just look for This Week in Science, also TWIS, TWIS. We live broadcast, live stream Wednesday nights, 8 p.m. Pacific time on YouTube, Facebook, and Twitch. We are TWIS Science on Twitch, Twitter, and Instagram. And if all of this is just too much to remember, head over to our website, twis.org, where you can find show notes, links to episodes, and links to subscribe on some of those big podcast platforms. But now, it's time for the science. So let's talk about Charon. It's not
Who?
Karen, it's Charon. We're going to talk about Charon and that red cap that she wears. I mean, where did she get it? Where did Charon get that red cap?
Who's Charon?
Who is
Well,
Hat?
Charon, or a moon of Pluto.
Ah.
Yes.
She
Pluto's
spells it
moon,
with
Charon, has a nice, jaunty red cap at one of its poles. And the question is, where did this redness come from? We first learned of its presence way back when, in like 2015, when the New Horizons mission passed by Pluto on its way out into our distant solar system. And the New Horizons mission took some really great pictures of Pluto and Charon and all these outer solar system objects, and everyone saw the data and everyone went, "Whoa! There's a red spot on the pole there, and where did that come from?" And so the hypothesis at the time, based on the evidence, was that because of the extreme swings of Pluto through its orbit around the sun, that there's some extreme seasonality, really, to Pluto and also its moons. So there's this idea that there is hydrogen that gets evaporated from Pluto, goes to Charon. Charon also has a little tiny atmosphere, and that this little atmosphere of Charon also gets kind of heated up. And there is a particular kind of radiation, this light scattering that takes place that they were thinking led to the creation of a red tinted methane product. So basically light impacting molecules in this itty bitty atmosphere as the atmosphere heats up and cools down, And so like whatever was in the atmosphere would kind of lighten up and expand and then condense and rain down onto the pole of the moon. And while in part this is correct, new data that they've been analyzing, they've determined, doing a simulation here on Earth of Charon's conditions, that it's not methane, it's ethane, which is interesting. Ethane is also a little bit less volatile. It doesn't really do a lot of stuff. And so nobody really knows where the red color comes from, because it doesn't make sense, because ethane is normally colorless and it doesn't normally get a red hue. So it's still a big question, but
So it's still the mystery.
it's still a mystery. Where did the
Nice.
red color come from? Now we have more of a molecular chemical pathway as to like what kind of processes are starting to get it to that point. How do you get the material there? How did it, what's going on there in the first place? And so we're getting a little bit better idea from the research in this study. And the researchers, they have published this in Science Advances, the Journal of Science Advances, and they are from the Southwest Research Institute, and they've used data from NASA's New Horizons mission with laboratory experiments doing, I love this word, exospheric modeling. Because it's atmospheres and things elsewhere, modeling other atmospheres on other things. It's fascinating. I mean, a little frozen moon on the last outpost of the solar system almost. It's got a little atmosphere, and it has ethane snow.
Yeah.
What?
It's got this red tint to it, and somebody
And the
looked
red
at it and
tint.
said, "Huh, wonder why that is."
So what I'm
Uh,
understanding is they have like basically aquariums, like fish aquariums in the lab. I mean, fancier than that. But then they
yeah,
put
fancier fish aquariums.
like
Yeah.
dry ice in it and then just see what color and then they pipe in different gas and see how it looks, right? I mean, that's what they're doing an artificial exosphere. That's basically all they're doing, right? Is they're just testing gas interactions and what they look like.
Yeah. Yeah,
Yeah.
but they're,
It's so
limiting
fun.
all of the, gas interactions, and then they also were using this. I mentioned the light
Right.
radiation, and so the impact of the photons, and so they also simulated this what's called the Lyman alpha glow to see how that would impact the chemical reactions and what would be happening. Yeah, so they have a special fancy frozen fish tank that
Yes.
they shine lights on. Yeah.
This is I feel like there will be science fair experiments that are baby versions of this, right? Where they like shine the
Possibly.
UV light through the fish tank full of gas. Like that seems like a very cool way to go. Find a gas that's reactive to different lights and then yeah.
I
Anyway.
love it. Who needs a science fair experiment next year?
Yeah.
You just came up with a great one.
Yep.
That's fantastic. Yeah. Anyway, Sharon in her red cap. We still don't know enough about it, so we're going to be waiting for all that juicy gossip as soon as the researchers can publish that. So, moving on, Justin,
What do we got
pea
here?
farming? You wanted to move on
Oh,
to farming, right? And what?
yeah. So this is actually
Snap peas?
ah,
Sugar snap peas. English peas?
so there's a cowpea.
Oh, no.
Interestingly, is not the pea fertilizer story. It's the
Okay.
cowpea mosaic is the virus in a later story. This is human pea.
Oh,
This is
urine.
a team of European and African researchers have been testing human pee as a potential fertilizer. So urine contains phosphorus, nitrogen, potassium. These are the main ingredients of a fertilizer. We had a story we were talking about a while ago where they were working with training cows to pee over a grate to collect their urine. Same sort of idea. You could use it as fertilizer.
Train
But why
humans
are
to
we?
pee over a grate.
Yeah, why aren't we already using our pee to grow crops? Is the big question. If we have this fertilizer that we have readily available, every one of us every day. Well, for one reason, there isn't any form of a pea to farm to table infrastructure in place. Also, oh no!
I mean, how many stories
I
have
gotta
we
wash my
had about
vegetables.
like medications and all the stuff that can be discovered from human urine? So there
Yeah.
limitations on what we can do here.
To be fair, that is only gross in the sense that in the best sense of I don't know where my food comes from or how it's grown, sort of a way. Because of course, cow poop, pig poop is a major source of fertilizer. It's not like we're just
Totally.
putting those seedlings down in sanitary earth and just watching them grow by the miracle of sunlight alone. No, they're normally grown in things that are fertilizers. Right? All right, so this is in their paper published in the journal Agronomy and Sustainable Development. The group described an experiment conducted with farmers in the Niger Republic and the use of human urine on crops. The researchers enlisted the assistance of a group of women living in an isolated part of Niger. These farmers have been struggling to find suitable fertilizers for their grain crops. It's high cost of the commercial products there and scarcity of large-scale livestock to produce manure. So, first rule of pea farming club apparently is don't talk about as pea farming. Don't call it pea farming.
Don't use those words.
Don't use the word. Well, apparently in Nigerian, the common word that you would use for urine is a curse word. It's considered a very offensive word. So they called it oga instead. Oga, which it has some other meaning, but they gave it this name. Anyway, it's still pee. Next, they separated the farmers into two groups. One that ran their farms the way they'd been doing things, and the other group that was going to do pea farming, going to be adding pea to the fertilizer. Over 2 growing seasons, crop yields were measured for both groups. The pea farmers, the pea was provided by the farmers themselves. So they know exactly what's going in there because they know maybe if you're on the antibiotics or something, maybe you don't add it to the mix, right? Maybe if you're on a medication or something, maybe you don't. But they were the ones providing the urine for their own crops. They were taught how to pasteurize, store, and dilute the urine for use as fertilizer. They also added small amounts of animal manure, which was less than the other group, but still. So it was augmented between the two. The data collected from the farms showed that those that had been fertilized using pea produced an average 30% more grain than the traditional farms. 2 years after the experiment, researchers found that more than 1,000 women farmers in the region were using their pea to fertilize their crops. They sanitized. They did have a sanitation system. Human urine is collected in small plastic containers, transferred to bigger containers, where it is stored for sanitation purposes at least 2 to 3 months before field application. And it was stored under a temperature range 20 to 24 degrees Celsius, which is basically the ambient temperature there. And that, according to a paper from 2013, leads to sanitation of sorts.
It's below body temperature. So anything that would have been potentially in the urine wouldn't be able to survive because it would supposedly be surviving in body temperature, but not below it.
Somebody says it burns the poor plants. Good question. So they did dilute. There was a dilution step, which I think was 1-to-1. Just add
it seems like just
1 cup of water for every 1 cup of pea.
It seems also, though, that we do know that the ground is like a filter, and also the sun kills bacteria and other things that can be dangerous. So, I don't know. It seems like putting the urine in the sun would be a good sanitation step as well. But maybe this is very fascinating to me. I want to know why it works.
And Justin, where was this again?
Nigeria.
So that's another thing I think is interesting is I want to say in a lot of countries besides the United States, people wash their fruits and veggies much more thoroughly than we do. They'll actually dunk them in diluted bleach a lot of times. And so I am curious about how that process worked and if that was part of the success of this process. Also because just the ick factor, right? Like if you know you're washing things that well, you might be less perturbed by it. Let's, not take a minute to think about in the United States that we don't wash our stuff super well. They're washed by the farms, but we don't wash them in our kitchen sink to the same extent, and we still have raccoons and feral cats and all sorts of things that are peeing all over our produce. So.
We do. Although not in your grocery bags, hopefully.
And that's where it's because this is fertilizer, so this is getting applied a couple times during the growing season and not onto the plants. It's not like to give the
au naturel application.
No, but I mean it's being applied basically to the soil.
Yeah.
It's for the roots of the plant before it grows the produce or before the grain, right? So it's not like you're spraying it on the grain. That would be too late for a fertilizer.
Right. Well, I'm all for anything that prevents us from putting urine in a bowl full of fresh water because that is a waste of water. So
Mhm.
if this can divert that, that would be great. Whoever came up with that, I'm mad at you. That was a bad
Oh,
idea.
I'm not.
You mean
I'm
letting
not.
it run
Whoever
out
that is,
to the river and tributary
that's the
system?
that's the
No,
greatest
I
invention.
feel like there are other things out there that we could be doing to reduce smell that wouldn't involve wasting a bunch of fresh water.
Yeah, but then
That's a
like
whole nother
wherever
thing.
you
Gray
are,
water,
there
recycling.
is,
Yes.
there is. But here's what you would have to do, Blair. You and probably all of the other residents on your block would have to leave your home each time, walk over to, because you got to have it some distance from the house, to the pit that was dug. That's got the nice little toilet sitting over the top
You're
of it.
telling me in 2022 there's not some sort of technology that we could come up with that would reduce the smell of urine, that would filter it on its way out, that wouldn't require gallons of water? Because I'm pretty sure there would be.
No,
That's just
well, there is, but again, then you're talking about the slide that has to be south facing, so with
Oh
the
my.
glass
Okay.
over it, so the sun can come and dry it. But it's
I think
still, you're
this
not
is
getting rid of the smell.
anything potentially sustainable and putting us in that direction that we can start thinking about putting on our radar for
Mhm.
future development.
But
It's great. So
gray water
gray
flushing
water.
would
Mhm.
make all the
Exciting.
sense in the world, and we don't do that enough. You're right.
Yeah. Oh, after all this water talk, though, I'm
Mhm.
thinking about maybe I need a cup of tea.
Yeah, would you like some arthropods with your tea?
No, what?
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 Researchers from Trier University and the Max Planck Institute for Evolutionary Biology wanted to look at eDNA in commercially sold tea and dried herbs. eDNA, that's environmental DNA. It's left behind in natural environments by creatures like spiders and other arthropods. And this is usually when you're talking about eDNA, you're not talking about crushed up actual arthropods. You are talking about their saliva, their leavings, speaking of pee, and poo. All this kind of stuff when they
The
crawl
frass.
on the frass, exactly. We try to give it a different name so it's less gross, right? So they crawl on these surfaces, they chew on these surfaces, they leave behind eDNA. And just like we talk about eDNA in the oceans from fish, that's also often from peeing. This is a similar measure. And so this was a kind of a proof of concept that they could test tea and herbs, because they know that the two best ways to study eDNA is in some sort of water or
Mhm.
in a preserved, super dry location. Those are the two best ways to get eDNA. So they went, what about tea and herbs? And they found eDNA for over 1,200 unique species of arthropods.
Wow.
Yes, this is in 40 tea and herb samples, just 40 samples, 1,200 species. They found evidence of 3,264 individual arthropods spanning very different types: predators, herbivores, detritivores, parasitoids. They found that every sample, if you picked one sample, it contained eDNA from at least 200 animals. And the one that had the most?
That's significant. What?
What kind of tea do you like to drink, Kiki?
I like black tea.
Okay, Justin?
The same.
Okay, you're
I'm
both safe.
I'm an Earl Grey girl.
Yeah, me too.
Green tea had the most. Green tea.
Oh, the bugs like the green tea, huh?
Yes, leafy.
You'd think there's a very bitter flavor to most green teas, though. You'd think that would be pushing the insects away, but
I don't know.
interesting.
So this research shows that the range of eDNA in studies can be expanded to pretty much anything that is preserved or dry. And so looking at actually commercially sold items could help them to look at the ecology of specific areas if you know where those things are sourced, all sorts of good stuff like that. And so this is a potential way of monitoring those specific ecosystems. Of course, this could provide useful as global warming continues. Does that eDNA change? Do the arthropods visiting that area change? And it can track other impacts like pollination and all sorts of other things. So eDNA from, in this case, food items can help keep a track on specific ecosystems.
Oh, that's interesting. Yeah,
Mhm.
that's a wonderful application.
Yeah. So think about
Huh.
that next time you're having some tea.
Yeah.
I would love to find out what the different insects are using. I mean, this is their ecosystem, right? Even
Mhm.
though but most of these teas are, I mean, I don't know. Are the teas in monoculture? How are
Mhm.
the
Great question.
insects using them? So
Mm.
if teas are grown in more complex, diverse environments, which I know some growers do that, does that mean that there's a greater diversity of insects? If it's monoculture, is there a lower diversity? And then how many of these insects are biting and chewing on these plants,
Mhm.
which have a lot of these antioxidants, which are also, those are compounds that are meant to protect the plants and be, you
Mhm.
know, trying to get the insects to stop biting them. Um, and
Yeah.
then, yeah, and how many of them are just landing there and it's a place to put a cocoon or it's a
Mhm.
place to have, a little evening away from the kids. Uh,
And that's sort of how I would picture it because there's
yeah.
when you're drying all of this, it's not like there's an indoor factory sanitary floor lab where they've got these outside or in barns, and it's just throw all the leaves in there and out there and lay them out and let them just dry in the sun and the air.
Yeah.
So the
Well, a
bugs
lot
are
of,
going to be
yeah.
everywhere. Yeah.
Yeah, a lot of, I mean, that's the tea leaves, usually they are just dried out places, sometimes
Mhm.
they're Blair, you've seen the inside workings of the, what is it?
The celestial seasonings
The
plant.
celestial seasonings
Yes.
plant. So did you learn what they do with their herbs?
I mean, there were lots of conveyor belts. It was many years ago now, actually.
Yeah, it was a while
I do
ago.
remember going in the peppermint room because it was
Yes.
very strong. And I also remember being told that herbal tea is not truly tea because it does not include any tea leaves.
Hmm,
So
interesting.
chamomile is not tea. Sorry, everyone.
I'm still
Yeah, I
gonna
guess
call
it's
it, it's
one
an
of the
infusion.
There's still bug DNA in it, though.
Yeah.
Guaranteed.
And, so Goldizator over in Twitch is asking about, the aging of the tea broke down the DNA, and not any more than just any regular outdoor process would. So there's probably a lot that is not preserved, but not destroyed either.
Hmm.
Yeah, well, it's one food product that you can't wash in hot water before you hand it over to the public because
Right.
it's
Truth.
be
It would ruin the
ruin it.
tea.
Yeah.
Exactly. It would ruin the whole purpose. Moving on from P to T to meat. Let's talk about the meat that we eat and its alternatives. Have you ever wondered whether the protein, the meat that you eat is whether it's treated the same by your cells as regular meat? Like meat versus tofu versus wheat gluten versus, like, have ever wondered whether these different proteins get the same treatment
Hmm.
from cells or whether
From,
there's any different treatment?
so I would assume that my veggie burger is going to be as useful as if I'd eaten vegetables and not meat. So
But
there's
that's a
probably
veggie burger. That's not an impossible burger. That's
or a
different.
fungal
Which is
or
a meat
a
alternative, right?
Right.
fungal
Something that is
burger.
specifically
Yeah,
supposed to be a chicken substitute, for instance.
Because
right. So
there's protein in mushrooms, so
Mhm.
that's a question. If you eat a portobello mushroom, is that considered the same by your body as the grams of protein that you would get from a teeny tiny bit of steak? Which my guess would have been that the difference is in the type of other nutrients you absorb at the same
yeah,
time. Right. And
yeah.
so, like, you talk about nutrition, you say you need a little fat to help with the processing of protein, and you need omega-3s, and you need all these different kind of, you need fiber, you need all these things in concert to get the optimum benefits of these items. But I've never really thought about whether, if you're just looking at protein, if it would
Right.
matter where it came from.
That's
Well, some chemists decided to check on this, and they are publishing in the American Chemical Society's Journal of Agricultural and Food Chemistry their analysis of how cells basically absorbed and dealt with chicken protein or a meat substitute that they created out of soy and wheat gluten. When they took this mixture, that was not a commercial on the market kind of mixture. This is something that they created. They created a model meat alternative out of these products. And then they used an extrusion process. So it ended up being this kind of long, fibrous, filamentous meat substitute, very similar to chicken, very similar to muscle cells and the way that they are situated within the muscle, long and fibrous. Anyway, they cooked them and they cooked pieces of chicken. They then ground up the meat substitute and they ground up the chicken. And then they put that ground up chicken and then the ground up meat substitute in an enzyme that humans use to digest food. So an enzyme
Oh,
that's
no one's
found in our...
eating these. Okay.
They weren't eating them. No.
As you kept
No,
going,
this
I was
is
like,
a-
that sounds less and less yummy.
It wasn't
So
very...
it's a food science lab,
They-
so they've
well,
got
in a lab,
they've
they
got
created
a
the
faux
thing.
digestive system.
Okay,
Yeah.
all right. I'm with you. I got it.
that they do
Simulated
chemical analyze
digestion.
after.
Yes. So they simulated digestion in a lab, and then they basically they put these digested meat solutions in with cells in petri dishes, and they determined that the meat substitute proteins were not absorbed as well as the chicken proteins by the human cells. And so the question is, what's going on there? And so, Blair, I'm to your point earlier about how things are put together. All these researchers did is take soy and wheat gluten and create a meat-like product that doesn't go into all the other stuff. What about fats? What
Mhm.
about the other nutrients that are in chicken? What other stuff is in a slice of chicken
Mhm.
that would make the cells more likely to incorporate them, to endocytose them? What would make those proteins more useful? When you would think a protein is a protein is a protein, right? But they're not. So what is the difference, and how? Obviously there is a difference.
Yeah, my
Just
money's
the-
on that. That it's
Yeah.
like the stuff in the chicken.
Yeah. So that, but
Yeah.
you know, we don't know. So is it stuff in the chicken? Is it some aspect of the initial protein structure, and then how the different proteins cook because of the amount of water and fat that was in the different substances that led to the eventual shape of the proteins that made them less or more likely to be absorbed? But this is a study. I don't know, but it's not exactly the same as having your fake chicken nugget versus a real chicken nugget. I mean, it's not exactly the same as that, but we're not there yet with that understanding. But this at least is an interesting piece of the puzzle.
Yeah.
What makes the fake meat
Mhm.
different from the real meat?
Yeah, what would really make that interesting is if we found out it was like the fat, right? And then one of the things that fake meat is supposed to be promoted for is that it's healthier for you. So if we have to make the fake meat less healthy in order for the protein to be better for your body,
well, yeah, more useful.
my
Yeah.
God, what are we doing?
What are we doing, people?
I'll tell you one thing, that fake meat,
Mhm.
there's not a whole lot of environmental DNA from insects.
At that point, probably not.
The
Although
chicken, but maybe the soy has it.
Although I bet you it does have environmental DNA from whatever organism was producing it.
Yes, certainly. Has normal DNA.
That too.
I don't know.
That too.
But moving on, we want to be healthy, right? We want to live long lives. So should I just stand on one leg?
Yep.
Is that
Well,
just-
you
just put
scooped
it
the
in?
whole story.
I'm doing it right now.
You scooped the whole story. That was my
You'd
big reveal at the
never
end. I was
know.
going to do all this lead up, and then it was going to be stand on one leg, and that was the big ending. Well, anyway, this is good news for people who like to stand on one leg. Because that ability to stand on one leg for just 10 seconds cuts in half the risk of death from all causes, according to research published
Kiki's
in
showing
the
off.
British Journal Sports Medicine, suggesting that a simple, safe balance test could be included in routine health checks. Oh, I hope not. They've got so many ridiculous things. When you
I'm
got
doing
okay,
it
so
doing
the
the giggle pose. Sorry.
hey, I
I'm
got a
doing my
question.
yoga now.
Yeah,
How do they? Oh yeah, no, yoga in a chair. You're just gonna
I'm standing.
wrap it up like this, and then with one leg.
All right. Okay, so what's going on with this
So
story? What does this mean?
they're gonna make people stand on one leg now to see if they're healthy. Which you don't turn your head and cough to check for hernias anymore. I don't think, but it's a thing guys had to do. They would grab you. And they'd say, okay, and they'd turn your head and cough. And the reason you did that is so you didn't cough on the doctor. But they were looking for hernias. Like, there's other ways to look for hernias. When they listen to your heart, do you know how many doctors can actually pick out a heart murmur or an arrhythmia just listening to your heart? It's none. They can listen to your lungs just fine, but they're not that good. Your doctor's not trained that well. They do a lot of things. Now they're gonna have people standing on one leg, falling down. Well, at least they're already at the doctors. So
Doctors listening to TWIS, direct your emails to Justin.
Well, actually, no, but I think actually doctors are why we don't do that anymore. Like, it was this, we had all these weird practices that doctors would do in a checkup that at some point they're like, I've never found a hernia. I've been practicing medicine for 30 years, like making people turn their head and cough while I grabbed them by their genitalia. Didn't find one. Anyway, no hernias that way.
But
Anyway,
why is this going to be more successful
trying to
than
get
the
to that.
coughing?
I keep trying
This
to
is
get to
going to be
that.
more successful. Why?
I don't think it is. Unlike aerobic fitness, muscle strength, flexibility, which wane quickly as we age, balance actually tends to stick around reasonably well until about the 6th decade of life. Then it starts to wane. Oh boy, does it wane rapidly, say the researchers. Balance assessment isn't routinely included in middle age and older folks, so there's no standardized test. So they kind of created a standardized test for this. This was all set up back in 1994 to assess associations between various measures of physical fitness. This is part of the ClinMEX exercise cohort study. The one-legged study included 1,702 of those participants, aged 51 to 75, with an average age of 61. As part of this checkup, participants were asked to stand on one leg for 10 seconds without any additional support. And they had to keep their arms at their sides, look forward, and I think they had to have the free leg behind the calf of their other leg.
Oh.
So they couldn't do bow pose. Like I can go out there and bow pose and hold it probably longer than I can do that weird posture that they're talking about.
I'm trying to put my leg behind my other leg.
Yeah,
I have my arms down.
Yeah,
It's
at
a
least
very-
let me go flamingo. Come on.
Yeah, no, that would be, but they said because they need everybody to be able to do it, they tried to make it as simple of a one-legged test. And in fact, you could pick a leg and you had 3 attempts on either foot until you could get to 10 seconds, and then it was a pass-fail. If you just couldn't do it in any of those attempts, it was a fail. If you even got 1 out of 3, you passed the thing. All in all, those people who were, let's see, what is it? 51 to 55, or about 5% couldn't do it. Mm-hmm. In the next 5 years, it was 8%. Between 61 and 65, 18% suddenly could not accomplish this task. Just under 37% amongst those 66 to 70 years old. And over half, 54% of those aged 71 to 75 were unable to complete the test. When they looked over their data and stuff, what they found was the chances of dying of any cause. You get cancer, people died of heart congestion problems, they got the COVID, hit by a bus, drowned while snorkeling off the coast of Hawaii. However, they died. The chances of death, the 10 years after failing that test,
Mhm.
increased by 80 something percent. Oh, I lost my number.
Yeah, but
84%.
so it increases massively if you can't stand on one leg for 10 seconds.
Yeah. So when I first read this, I was like, well, of course, the 75-year-olds are the ones doing the worst at this test. So you look at all the
If they're gonna be
numbers, and
living to 85 or yeah,
right,
yeah.
the chances of making their next 10 years much tougher than the 55-year-old making his next 10 years, right? So that seemed weighted, but they, according to this, they waited for all the factors they could, including age, and they still got to 84%.
Mm.
Yeah, so it's probably, related to neurological factors.
Mhm.
It's probably related. I mean, there's probably just general health factors involved as well.
Yeah, in general, it says here
Mm,
those who failed the test had poor health, higher proportion
mhm,
were obese and or had heart disease, high blood pressure, and unhealthy blood fat profiles, as well as diabetes was common.
mhm.
38% of those who couldn't do it had type 2 diabetes. So they were 3 times more likely, that group. But again, that's something that can also hit you harder or hit you later in years.
Right.
So, but this reminds me, there was another study like this that I'm cognizant of. And every time I'm walking around now, there was one almost exactly like this. And I'm wondering if they used the same data because it was the same like chance to die in the next 10 years of any cause, which was the rate that people walk at, how fast you walk, was related to how much time you had
Mhm.
left. So the slow walkers they found died quicker than the fast walkers. Fast walkers, it means you got a lot of life left in you. So if you can walk fast and stand on one leg for 10 seconds, you're fine. No need to go to the
You're
doctor no matter what
great.
they.
Yeah. Yeah. Well, I mean, this just it really does reiterate the importance of doing whatever you can do to maintain the best health that is possible for you
Mhm.
and maintaining activity as late in life as is possible, just as much as you can get will help with that general health. And yeah, test yourself every day if you can. Stand on one leg. Make sure you can do
I
it for 10 seconds. Sit
can't
down in a
believe
chair, stand up, stand on one leg. There you go.
can't believe old man Jackson passed. You know, he was such a fast walker and he could stand on one leg. Yeah, but he drank heavily and smoked a carton of cigarettes a day.
Right.
Oh yeah, but did you see him move around? He
But
was
he
quick.
can stand on one leg better than a flamingo.
Yeah.
Oh
That's
my goodness.
amazing.
Well, as we're on the being healthy tip, one thing that
Mhm.
you can do to be healthy for yourself can also help the climate. A new study published in Nature Food estimates the carbon footprint of the global food transport system. And it's a fascinating analysis of emissions from the entire food supply chain. So they're looking at emissions from the transport of fertilizers, like the animal fertilizers we were talking earlier, machinery, the feed for animals like the cattle that become our food, and the moving of food itself. And they found that in 1 year, global food miles were responsible for 3 billion tons of carbon dioxide equivalent emissions. So that includes carbon dioxide and other greenhouse compounds that have effects. So 3 billion tons. And this is larger, significantly larger than previous estimates. Now, there are some people who are still debating, as always happens in science, debating the estimates that have been made in this study because uh, some. Uh, some people contend that they are overestimating certain aspects of the emissions and how much fuel is used, like for train transport of coal. For instance, in the United States, coal is transported on trains very often and not on trucks. So trains are going to be a little bit better on the emissions than trucks on the road are. Slightly. Anyway, there are all sorts of details, and it gets very complex when you really dig into the food systems of our planet and the global supply chains and how we use them. One thing that I thought was very interesting was their attempt to look at for instance, the entire supply chain of something like red meat for the country of China. Chemicals for fertilizers for the feed for the cattle are produced in Canada and in North Africa. Those go to support the growth of vegetables and fruit and soybeans in the United States and Brazil. Additionally, those are then transported to China, where the livestock are grown to be able to feed the livestock there. Coal is produced in Australia and Indonesia that is transported to China for to power the manufacturing process for the sale of the red meat to the Chinese citizen. So it's an incredibly long process, and each aspect of the process has own inputs and outputs. So the final point to this story, however, is not just, oh, let's all be sad about all the difficulty of the human food system and the web that we have created for ourselves, but rather we can take a picture of how food and energy are emitted and used, or food is used and energy emitted, the carbon dioxide is emitted uh, around the globe. And what it tells us is that plants are more often grown and shipped around the world. And plants can be something that can be grown more locally and have less of an impact on these carbon emissions. And especially richer countries, if we reduce as we've said lots of times, our reliance on meat eating, that would reduce a big aspect of these emissions. But not just eat more plants, but eat more plants grown locally. So eat more seasonal produce. Don't buy things from Brazil, or even though this is a global economy and we have the money and we can buy whatever we want whenever we want, if you start shopping from a more local seasonal mindset, it has a huge benefit, or it will have if at the population level, people make choices like this to be able to shop locally.
The more you buy local, also the longer the food lasts,
Yes.
which reduces contributions to food waste, which also reduces the need to buy more food, which also feeds this whole system. So that's, yeah. Is that the chime of being correct? I just got that.
Da-da-da-da, you are correct.
Winner. Yeah, so there's that. There's also, it's if you're shipping food, there's a better chance it's being preserved in some ways, which refrigeration I think is the number one cause of carbon emissions according to Project Drawdown. So
Mhm.
that is a thing.
Huge. Yeah,
So
refrigeration,
there's
that's the big part of the emissions is the refrigerated trucks for produce.
mhm.
It's a
Yeah.
big deal.
Yeah, that's something that I was not aware of. This whole global food shipping, or you're talking about food lasting longer. Now I'm here in Denmark, which has plenty of farming, but not that much.
Yeah.
A lot of produce comes from elsewhere in Europe, and you
Or
have to
even
eat it the day
Africa.
you buy it. You have to
Yeah.
eat it the day you buy it, or you're going to throw it away. It's that fast.
Yeah, so I mean, next time you go out to the grocery store too, look at the little tags on your fruit that you buy and see where they're from, because you might start to notice, okay, if I go to this store, they have a lot of food. In California, there's some stores you can go to where it's all from local farms, and there's some stores you can go to that like to wrap their vegetables in plastic, and they're often from Mexico, and so they have to travel a lot farther. And so, just it's something to educate yourself about so you can make good decisions.
Yeah. And yeah, just know that the carbon footprint of the more distantly traveled foods is going to be higher than something local,
Buy
more
local.
likely. Yeah. It's complex, though. It's a very complex system. But yeah. Like, just that the old saying, "Think globally, act locally," it just keeps coming back to it's a very accurate statement. Alright, Blair, you had
Mhm.
one more story
Oh,
for this
I'm
first part of the
so
show.
excited. Are
I
you all ready? Everyone get snuggled down in your bed. Pull up
No!
your covers. Get your eye mask just right on your forehead, ready to come down. Put
No!
on your white noise machine, and I want to tell you about your face mites
Oh gosh.
that like to come out at night on your face.
It's
Well,
nighttime, and the mites are
Yeah.
crawling out of the pores in your skin and
Mhm.
crawling across your face so that they can mate with the other mites, and they're eating your dead skin cells and oh,
Yes.
Demodex, we love the face
Oh,
mites.
face mites. They're passed
Face
on
mites.
during birth. Did you know that? They're
What?
carried by almost every human. They measure around 0.3 millimeters. They're found in
Tiny.
hair follicles, on your face, and in the nipples, and on your eyelashes. That's why you're always supposed to wash off your face makeup. For those of you that wear eyelash makeup, you're supposed to wash it off before you go to bed because the face mites like to eat the makeup. Then they also mostly eat sebum, which I had to look up. Sebum,
Sebaceous secretions?
exactly. So sebum, as in sebaceous glands, as in sweat. It's oily, waxy substance produced by your sebaceous glands. And so these little buddies, these little co-pilots on your body that are always with you, somebody did some DNA sequencing on these little friends, and they found that they're actually changing. They're becoming so simplified organisms that they may soon become one with us.
Ooh, are
Yes.
they
the
still gonna
mites
crawl
move
around on our skins?
in, but they
Mhm.
never move out.
Exactly. So this is the first ever genome sequencing study on D. folliculium, folliculorum. Excuse me, I missed an R in there. D. folliculorum.
Folliculi,
Yeah. Yes, there
follicula,
you go.
folliculorulum.
So they found that their isolated existence on us and that we are passed down from birth means there's inbreeding going on with face mites. We're going to make them even more appetizing to talk about the fact that they're inbreeding. They're causing them to shed unnecessary genes. They're narrowing down their genome so much that they are transitioning from external parasites into internal symbionts. They found these mites have a different arrangement of body part genes to other similar species, which is most likely due to them adapting to the sheltered life inside our pores. We are giving them such a good life that they are throwing away unneeded genes. They found the following. They're extremely simple organisms with tiny legs powered just by 3 single cell muscles. 3 cell muscles for their legs. They lack UV protection, which is why they have to count at night. And they have lost the gene that causes them to be awakened by daylight, why they're up at night. They are unable to produce
Oh, wait.
melatonin.
Say that again.
So they lack UV protection.
Yeah.
They have lost the gene that causes them to be awakened by daylight.
So is that I wonder if that's also helping so that we don't feel them crawling around when we wake up. Like they wait
We
till
wouldn't
we're
be able to
asleep
feel them.
before they're like,
They're too
okay,
small.
humans asleep, let's party.
No. You wouldn't be able to feel them. They've also been left unable to produce melatonin, and which is how many small invertebrates are active at night. But they fuel their nocturnal activities by using the melatonin we secrete in our human skin at dusk. They're using us as the pharmacy.
Oh, we
They're-
give them their wake-up power.
If
Yeah.
I've been taking extra melatonin lately, does that
You're
mean
feeding them.
Yes. You're making them have a disco on your face.
Oh.
Their
Boy, that's gonna help
reproductive
me sleep.
organs. Come on now. We got to talk about the mites' reproductive organs. They have moved interiorly. So the males have a penis that protrudes upwards from the front of their body, which means they have to position themselves under the female when mating. And they copulate. I want you all to picture this now while you're snuggled into bed, as they both cling on to the human hair. One of their genes has fully inverted, giving them a particular arrangement of mouth appendages which protrude extra for gathering food. That's just for fun. The lack of exposure to potential mates that could add new genes have set them on this course for what may also be an evolutionary dead end. We might lose our face mites.
No.
If we do, this is
I
actually, I
support
want to
my
talk about
face
this
mites.
for a second. Yes.
But yeah.
We don't know what will happen if we lose our face mites. Are they keeping something at bay by occupying our follicles? We don't know. Are
So
they
what I
cleaning us?
wonder, like, is there, are they keeping down bacterial infections? Are they, are people less likely or more likely to have acne if they do or don't have face mites? I want to know the answers to these questions. If
Yeah.
I am wearing sunscreen, what is that doing to my face mites? If I am going to the spa and having acid peels, what does that do to my face mites?
It's
I
a
want
great
to know.
question. Yeah, and speaking of healthy skin, apparently some people like to blame their face mites, not me, but some people like to blame them for skin inflammation because there is this theory that they don't have an anus, and so they actually just accumulate feces throughout their lifetimes, and then
Yeah.
they just kind of explode when they die.
Yeah, that's not true.
No, the DNA tells us that they do have an anus, and so they have been unfairly blamed for these
DNA
skin
says.
conditions. Thank you,
That's
DNA
awesome.
analysis.
Because
Yes.
that's like the one thing I remembered
mites have
about
an anus.
face mites is that they never went poop. They just ate until one day they just they burst.
It's
Yes.
not true.
Oh, well, that's good to know.
They're just
So
pooping
wait,
all over your
where's it
face all the
going?
time.
Ah,
Yeah.
that was
But
not
it's
really
just
better.
Tiny bits.
Not really
tiny
better.
bits. It's better.
I mean, perhaps they are leaving their frass. Would it be frass if they're a face mite? I guess
I don't
if they're an
know.
insect. I don't know. Maybe they're leaving them in the hair follicles, and that is leading to acne and inflammation. Maybe that's where they relieve themselves. I'm gonna guess they don't control it, and it's just kind of whenever. They don't seem to be that complex of an organism.
No.
No.
Yeah, they have a midden like a rhino. Like, this one
right
pore.
this spot right over here.
Yeah.
That's my face mite midden.
No. Exactly. Yeah. So anyway, face mites, we've done the DNA analysis. They are complicated, but getting less so. And I do hope that they don't go extinct. You hear that, buddies? Just here we go.
I'm gonna wash my face and
Are you
take
sure
off
you
my
can't feel
eyeliner,
them?
but I do hope that I do it in a nice way. Keep my face mites happy and healthy. I want a happy skin ecosystem. That's what I want.
I can feel my face mites now. But it's sort of like paying attention to your own breathing. Like now that you focus on it, and then now I have to control the breathing. This right now, I can feel the face mites.
Great. So I'm going to add to that by telling you I looked up what Kiki was kind of asking about. They are arthropods. They are, in fact, arachnids. So they're cousins of spiders and ticks. You're welcome. Enjoy. Teeny tiny spiders on your face.
Little tiny spiders that live in your face.
Or ticks, even worse.
Oh, they're mites. They're face mites. Oh, everyone.
Now I gotta find a kind of bug that I can put on my face that'll eat them.
No.
And then I'll have to find another kind to put until I get one big enough that I can see and just. And then
I really do want to know so much more about face mites and their role in the face. And there are so many questions. What is Derek
usually
Schmidt is asking?
Person. I don't want to know any more about it. I want to never hear about it again and forget that I learned
Well,
something.
that's too bad, Justin. If you're going to keep being on this show, I will bring them up again.
Like, is this why? I mean, washing your pillowcase. Are you washing away the dead face mites when you sleep on your stomach, on your face? Like, what happens to the face mites that are on the pillow side versus the not pillow side? I'm so curious.
Are they anywhere but the face? Do they just stay to the face?
Yeah,
are they,
I said
whole
the
body?
nipples is the other place.
The nipple,
yeah.
right? But they're not like. Are they like also all on the arms and everywhere else? Because if they're everywhere, you're just kind of.
My understanding is they congregate. They congregate in certain areas.
Well, the face has the best view.
Yeah.
Hey, I'm taking a ride. It's like riding on the top of the Statue of Liberty as she walks across New York City.
Now I feel like a giant spaceship.
Yeah,
You
kind
are.
of
you are
animatronic robot. Kind of like
Spaceship human. Everyone, might you enjoy This Week in Science? We hope that you're enjoying the show this week. This is This Week in Science. If you are enjoying it, please take a moment to subscribe. Head over to TWIS.org and click on the subscription links on our page there, or head to your favorite podcast provider and look for This Week in Science. All right, we're going to come back to a wonderful part of the show that we love. Not the COVID part of the show. Oh, no. We're skipping that one today because, everybody's skipping it these days, even though everybody's getting it. But anyway, it's time for Blair's Animal Corner.
with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh my gosh, I am so honored to bring you tonight the story of teeny tiny frogs that are bad at jumping. This is a story about Brachycephalus, which is a genus of frogs. They are called pumpkin toadlets, some of them, and some of them are called flea toads. They have become so small that they're no longer able to maintain balance during quick maneuvers like jumping. They're also just really bad at walking in general.
Oh, they'll all be dead in 10 years.
That's right. They have
Maybe.
no balance.
Maybe. There's a reason evolution has done this, which is part of what we're going to talk about. But so they lose control of their trajectory mid-leap. It starts like it looks like it's going okay, and then it just starts to go bad. Here, we're going to watch a video. And so they're kind of puffing a little straw
No. Oh,
at these
no.
frogs, and it causes them to jump. And for almost half of it, you're like, "Oh yeah, you got it," and then things just go terribly wrong. I can't.
So is
even...
this?
I need
That's
to play
really
that
bad.
over again.
Is this a hybrid by any chance?
No. So this is a whole genus. There's a bunch of species. You're going to see a bunch of different species in these videos.
Okay.
And so they're all just terrible at jumping.
Oh, gosh.
It's like they freeze as
Yes,
if they
exactly.
they leap and then get to the full extension with their legs straight and their arms out. Their body is extended, and then they get stuck.
Mhm.
And then they just tumble head over tail until they land however they land. That's
It's like if a cat fell off of a dresser, but instead of like contorting to land, it just stiffened. And
Yes, exactly. Yeah. And so I guess the idea is they really don't try to jump as a defense mechanism. It's a last resort for them. And so if they're actually jumping, they're just like, I hope this works out. And so they're just going, they freeze up, they're going for it.
No, it's the freeze game. They got a buddy you can't see off camera there, who as soon as he jumps, they go freeze. And he's like,
and they land on their legs, and their legs don't bend, and their balance is all off. They're like it's like a not fight or flight. It's the freeze response.
Yes, exactly. Yeah. There should be 4 videos, so there's some other ones.
I'll go find them.
Of some other just poor examples of jumping.
So there's this isn't just this one frog. It's not a hybrid, because I kept thinking it's got to be a hybrid. It's like one frog that had a big upper body, one frog that had different legs, and then they got a hybrid of the two, and now it just jumps wrong.
Yes, here's another one. Oh no! Oh!
How does it even exist? How did it get
So
this
that's
far?
what I'm going to tell you.
That's what I'm curious about.
Yes. So the reason they exist.
I was hoping.
I'm so sorry. If you are listening to this as a podcast,
you need to look for these frog videos.
If you're driving, pull over and look this up right now. Go to the show notes, go to twist.org, look at the latest broadcast, click on the link, look at these videos because...
Did you say the latest Frogcast?
Yes, the latest broadcast. If I didn't, I wish I did. But okay, so the reason they are so bad at jumping is that they are small. They are so small. They have gotten so small that they are no longer able to maintain balance. How does that work? They looked at their vestibular system. This was a 4-year initiative among 18 institutions. They looked at 3D models from CT scans of more than 20,000 museum specimens of this genus across the United States. They stitched together all these different frog scans to get as close as they could to a comprehensive model of their vestibular system. The vestibular system is a fluid-filled network of spiraling chambers inside the inner ears. It looks kind of like a nautilus. And when it moves, when we move around, the liquid moves along with it because, you know, gravity. And that brushes against these tiny hairs that send electrical impulses to the brain that tell us where our orientation is in space and how to maintain balance. So kind of related to the whole one-legged conversation we were having earlier. Sometimes people's vestibular system starts to go bad as they get older, and that can have to do with their balance issue. So anyway, the vestibular system is the main mechanism how most vertebrates are able to do the acrobatic things that they do. What scientists have discovered is that there is a specific restraint on the size of a vestibular system to allow function. For example, baleen whales have vestibular systems. Their chambers are only slightly bigger than ours, even though they're huge compared to us. They don't need to be that much bigger. They might not function if they get too big. Similarly, the chambers within the skulls of these toadlets are as large as they can possibly be in that small head. They look like overinflated balloons on these scans. So they are as big as they can possibly be relative to their heads. But even in that case, it's not big enough. The vestibular system does not function enough for them to maintain balance when they jump.
Because they dwarfismed down to the point where now I get it. Okay. So there was a bigger version of this frog, did just fine throughout evolved, could do the thing, and then I don't know, they got isolated on an island or however it happens. Dwarfism took over.
So they don't really think this is island dwarfism. They think this is actually specifically an evolutionary tactic, and it is because they are so small, they go unnoticed. And so the benefit of being small outweighs the benefit of a speedy getaway. So basically, they're more likely to just go unnoticed than have to run away from predators, and therefore, it is more important to be small than to be graceful in this case. So I will also throw out there, go ahead.
I was just going to say, and that it's just a huge evolutionary benefit. The size has allowed them to survive, whereas their vestibular system would not. If they were if they were continuing to be predated upon heavily, then they would have been eaten because they wouldn't be able to jump to get away.
Right.
So so this makes sense. Yeah. So it's really just the vestibular bones and the whole system's gotten so small, there's just no space for anything inside of it.
Right.
It's just a little thing. It's a little nubbin that doesn't work it doesn't do nubbin.
Yes, and MT in the chat room in YouTube brings up a really good point. They still have a jump reflex or instinct. Yes. Yeah. So they still have this instinct to jump, to get away. That is still their best case. If they've been spotted, they can take a tumble and maybe kind of right themselves and hide again, and they'll do okay. And so who knows, maybe eventually if they keep getting smaller, they won't even be able to jump anymore. I don't know. But just to add to the craziness of these frogs, also just FYI, they can't swim. They don't have tadpoles. They don't get around much. When they've monitored acoustic behavior of them, they record the same individual at the same spot over the course of a year. They really don't move around. They find a good hiding place. They stay there. And in 2019, I do believe we reported on it on this show, they were found to have fluorescent bones.
Oh, it's the same
Whoa!
guys. Okay.
Yeah.
Fluorescent
Yeah.
bones.
So these
You
guys are
guys
just
are
a barrel of
rad.
fun. Yeah.
I got
They're just
fluorescent bones, but I can't jump.
no. So
Fascinating. And
anyway,
they don't have tadpoles. I thought that was just
no.
kind of an obligate frog thing.
No, there
Fascinating.
are a few kinds of frogs that will go straight to froglet.
Wow. Okay.
Yeah.
Probably smaller, more likely to go straight to froglet. But
Mm-hmm.
yeah, fascinating.
Yep. Yeah. So there's the awkward frogs that are still winning at life by being very, very small. And now from frogs to frog-eating bats.
Woo! Let's go.
Yes, I want to tell you about researchers who train bats to associate a ringtone with food. What they found was the bats remembered the ringtone for up to 4 years after having been released in the wild. This is, as far as researchers know, the first long-term memory study involving animals being released into the wild, recollected, and then tested again. So there's been lots of studies on long-term memory: sea lions for 10 years, tortoises for 9 years, dolphins for 20 years, but those were all animals that lived in a lab or in captivity the entire time. So this was an important study because they taught them this association, released them, and then brought them back. And they used something to teach them on that they didn't interact with, most likely, in the wild for the entire time they were away from it. So they really did remember this kind of in isolation. So the way that they did that is they took frog-eating bats from the wild. They did a series of cognition tests where they were exposed to an attractive sound, which in this case was the mating call of the male Tungara frog, which is one of the things they love to eat. They fluted that sound and they were rewarded with a piece of food. And then over time, they mingled that sound and gradually replaced it by a ringtone. But they still gave them a reward. So then by the end of the study, they were able to introduce other ringtones, but they specified this ringtone means food, and the bats responded, and they were able to discern the differences in the ringtone.
That would get annoying after a while.
Yes, although I
Poor
do
bats.
wonder if any kind of lost researcher wandered out into the forest and they started getting text messages, "Where are you? We can't find you." And then they just started getting hit in the face by bats. Where's my food? So once they learned the ringtone, they were microchipped and returned to the wild. Then they collected them at a year, 2 years, 3 years, 4 years, and they were able to test them on their retention of that ringtone. And so it was successful. They found that 4 years, even though that's a long time to hold on to information you might never use again, 4 years was enough for them to retain that useful information in that one instance and be able to associate it with food. So this is beneficial for a bunch of reasons. Obviously, it has an impact on wildlife conservation and reintroductions to the wild. It's a reminder that whatever you kind of teach them when they're in captivity and what they learn to associate, that could carry on with them for a very long time. Mm-hmm. But it could also impact future studies. This is a really cool proof of kind of the mechanism that we could use with other animals in the future. And it shows that it's possible. If these bats were able to retain these memories for this many years, then that is a motivation to try it with other species as well.
Yeah, I mean, this is the big question for longer lived species. How long do they retain the information? We have this long held assumption that, oh no, they're not going to remember any of this stuff, or we just, haven't really tested these things.
Yeah.
Yeah, and those number of years that those different creatures, the sea lion 10, the dolphin 20, those like they tried it 11 years and it's like, sea lion forgot. Or is it that the study was, they tested it out to 10 years and it could go beyond that?
Yeah, see, that's the question. Yeah.
Yeah.
And it depends on the study. Some studies, they do find kind of the threshold, and some studies just end.
Right.
And so it's
we did it 20 years
at
apart.
least
They
as
said.
long as, I mean, you
Yeah.
know, in this case, 4 years, it's a suspiciously similar number to how long it takes to get a PhD.
Yeah. It is. Yeah.
Suspicious.
Huh. Anyway, something
We'll
to
be
consider.
expecting to see another paper out of this in another 4 years or so after
Mhm.
another student picks up the project.
Yes, yes. Or the main
Longer.
researcher carries it on to their postdoc. Who knows?
Exactly.
Yes, so animal memory, another episode of animals are smarter than we gave them credit for.
It's an amazing long memory. I mean, long memory. It's good if you're long lived to have a long memory. That's going to help with survival, and it'll make, because it makes things easier. If you remember things, then you can discriminate between choices that need to be made. Yes. All right, Justin.
Uh-oh.
It's time for you to tell us about some
Let's see.
science.
What do we got here? So, well, what is it? It was about 100 years ago. On the suburban outskirts of Canterbury, Kent, in jolly old England there, workers unearthed some odd-looking artifacts, hand axes and other odd tools that they found while digging ditches back in the 20s. These artifacts were determined to be very old. So they sent them to a museum where they have been stored ever since.
Yeah, it belongs in a museum. I get it.
It looks very old. So you give it to the museum folks because that's what they handle all the very old stuff. Now researchers have gotten around to asking just how old are they? They dated the artifacts using infrared radiofluorescence, IR/RF dating, a technique which determines the point at which sands of grain were last exposed to sunlight. So they went and they re-excavated the site, and they found more artifacts, and actually different artifacts, scraping tools and some other things that they hadn't originally found. And they have dated this to between 560 and 620,000 years ago. This is quoting Dr. Tobias Lauer from the University Tübingen in Germany. Tübingen? I don't know how to speak German. Dr. Tobias Lauer, who did the dating of the site. This is one of the wonderful things about this site in Kent. The artifacts are precisely where the ancient river placed them, meaning we can say with confidence that they were made before the river moved to a different area of the valley. So these are artifacts in situ. They were in a river. They got tumbled along by an ancient river. That ancient river isn't there anymore, so they at least can, get some of their dating that way. That gives you an older than date to start with. So you have also something that you can help verify or compare to your instrument data, your IR/RF in this case. So you also have something that another data point or another way of looking at that. So anyway, this is Dr. Alistair Keeve, University of Cambridge, directed the excavation. The diversity of tools is fantastic. In the 1920s, the site produced some of the earliest hand axes ever discovered in Britain. Now, for the first time, we have found rare evidence of scraping and piercing implements at this very early age. I think this is Homo heidelbergensis, a hunter-gatherer who ate very diverse things. With the scrapers and these pokey things those look like they may have been for processing of animal carcasses that they think could have been used for clothing or even for housing. So, based on the number of tools and the type of activities that they estimate were taking place, it looks like life was good 600-ish thousand years ago in England. Now, this is before England's first Brexit. This is while England is still, I think, attached to the rest of Europe. There isn't water separating, so they didn't have to sail or swim across. They could have just walked there. Dr. Thomas Prophet from the Max Planck Institute of Evolutionary Anthropology analyzed the artifact, said scrapers during the Paleolithic are often associated with animal hide preparation. Finding these artifacts may therefore suggest that people during this time were preparing animal hides, possibly for clothing or shelter. Yeah. So it is possible the European populations of Homo heidelbergensis evolved into Neanderthals, while a separate population of Homo heidelbergensis in Africa evolved into Homo sapiens. That is one of the theories. Well, really old, though, they're still older in England. They have a collection of footprints in Happisburgh, Norfolk, that date from 840 to 950,000 years ago. So things have been walking around there for quite some time.
Fascinating. Yeah, I mean, we always have thought about there's Homo sapiens and out of Africa, and okay, then there's Neanderthals, and okay, we were separated. Oh wait, no, we were living in the same places around the same. Oh, what are these other species? And now you're kind of off the cuff comment here. Oh, maybe hypothesizing that Heidelbergensis could have become Neanderthal. What? Like this? I'm like, what? This is awesome.
Yeah, it's all everything and it's all nothing.
Yes.
It's a soup. It's a stew. It's human stew.
How fantastic!
And that's why I'm skeptical that of that idea, the Homo heidelbergensis just becoming Neanderthal, and then the every information that we've gotten without fossils. Once we get a fossil, we learn a completely different story. That's the one thing they don't have here. This is stones that got maybe they were washing the axe and it ended up down the river.
Yeah.
These are lost tools into an ancient river. There's no skeletal remains anywhere. There's no jawbone or tooth or anything to tell us who
it was.
We can speculate.
Who are you?
We can have an educated speculation. And one thing we've learned a lot about these educated speculations is it's weirder, man. It's always weirder than we.
It's weirder than you think it is. Yeah.
The history of mankind is weirder than we ever thought it was. And there's a new therapy to combat cancer. Could one day cure it. Actually, it's curing it already.
Wait, what?
In mice.
Say more. In mice.
In mice.
But this is another colon cancer cure. We're finding multiples of these now. I feel like these are just, nobody should die of these things. This is coming soon. A combination of a plant virus and an antibody that activates the immune system's natural killer cells is showing tremendous early results in mice, according to researchers at the University of California, San Diego. In mouse models of colon cancer, the combination therapy eliminated all tumors and prevented their reoccurrence when they tried to give the mice cancer again. And the immune system had learned well enough to still be on the lookout and attack the cancer cells. So their results, 100% survival.
Of the mice.
Yeah.
100%.
For infinity. They're still alive today.
They are not, probably.
They never alive. We must kill you to find out why.
Yeah.
Well, they survived the experiment, and then immediately after they were probably incinerated.
Yeah.
All right, so put that in your colonoscopy pipe and smoke it, because the disease is soon, I think, to be a thing of the past. Therapy also increased survival in mouse models of melanoma. The work is reported in a paper published in Nanoletters. Fun fact, Nanoletters is the only scientific journal which requires magnification to read, even if you have perfect vision. Really? So they tried it also with just this plant virus, and it had a positive effect, but it wasn't nearly as good as when they combined it with this antibody. So there's this by having this, and this is the cowpea mosaic virus, which is a plant virus which normally affects legumes, but is completely harmless to animals and humans. And the antibody is anti-4-1BB, which you probably never heard of. So the antibody has the ability to, well, between the two of them, they can actually bind. Okay, the whole problem with the natural killer cells, they're there. They're around the cancer. They find the cancer. They say, hey, there's cancer here. And they create an antibody by attacking it. And that antibody is supposed to go out and train the rest of the cells. But there's not enough of them. There's not a big enough concentration of them that the immune system, because the cancer is horribly, terribly afflicting of an illness as it is, it's not actually proliferating that many cells at any given time. It's not, you're not going to get the antibodies up to the level where you're training the immune system to go and kill it. So, right. And they can also bind. They also have a ability to bind some of those antibodies when they get discovered to the cancer cell, so that it doesn't even get out into the mix and get trained. Something between this virus and this antibody, they're able to invite. I guess the plant virus attracts other natural killer cells. And the antibody keeps it from sticking, so that the other antibody keep this sticking mechanism of the cancer at bay. So it gets out. It gets the message out is basically what happens. And what was also cool is that the immune system, once trained, had immunity memory. So they went back and tried to, after the mice, they gave them the cancer and the melanoma, and then the mice survived. They recovered, and they said, "Let's give it to them again." Oh, and they recovered again.
It's amazing. So once you prime the immune system to recognize the cancerous cells, then it's ready to do it. It's like, I can protect myself now.
It's almost like an inoculation.
Yeah, that's kind of his thing. It's like, I just go ahead and give everybody this thing now and train up the system.
Might be a little early, but yeah, that's a fascinating.
Well, you'd have to give everybody
if this can move quickly through the pipeline.
That would be pretty wild if, as a kid, like now you get your Tdap or whatever. Like as a kid in the future, you could get your anti-cancer series of shots as a child.
Well, there's one
never have to worry about it again.
HPV vaccine.
Yeah.
I mean,
right.
we don't call it a vaccine against a sexually transmitted disease. Call it a pre-cancer treatment.
Cancer vaccine.
Totally.
Vaccinating yourself against cancer is what you're doing.
Yeah.
Yes. Oh, this is amazing. Oh, the immune system is so interesting. Now I'm just like, this is such cancer ecosystems, the immune systems, they all work together. So in such a fascinating way. Let's talk about brains and things.
Yes.
What shall we discuss now? I would like to talk about, let's see, what story did I bring in here? Oh, I think I put the wrong link in my rundown. That's too bad. What I wanted to talk about is how your breath can be used to identify you before we start talking about some other things. Yes. What does your breath say about you? Well, a new study.
I love garlic. Oh, sorry.
Yeah, exactly.
Mine says coffee and cookies this morning.
Well, this new study out of Japan, they have been looking into ways that biometrics can be used to identify individuals. So we all know fingerprints. We know pictures of your pupil, that your pupil is like a fingerprint, even more unique than your fingerprints, in fact. People use teeth. They're starting to look at microbes. They've been starting to also look at the chemical compounds that are released from your sweat. And so these researchers said, wow, we can take a look at stuff that comes out of people's sweat, But these compounds that are unique to individuals are usually found in parts per billion. So they're really, really tiny amounts. And so they're hard to find. And they said, what's another thing that we could possibly use? And they went, oh, I know. And so they started measuring compounds that come out in people's breath. And, yeah, well, you might think, oh, all the stinky food and all the other things. They found 22 compounds that can be used to identify people. And so they stuck these compounds on a chip and they started testing them on people. And so far, it's a very small pilot study. They've been looking at like 20, 30 people. It hasn't gotten to the mass testing that it needs to be to really be useful. But at this point, as they have been developing this fingerprinting system based on breath, it has been over 97% accurate in identifying individuals. That said, all the individuals who were involved in this study had to fast for 6 hours before the study, before breathing on anything.
Okay, before you enter the safety room,
you can't have had anything to drink. You can't have had anything to eat. You can't.
I'm never getting in.
No, to get into the safe or to get into this like panic room or whatever, you can't get in. Don't eat for 6 hours, then come back.
I would like to get into my safe deposit box, please. Yeah. Go fast for 6 hours and come back later.
Is this all the microbiome? Is that the idea?
Not necessarily. These are volatile compounds, chemicals that are released from, it's in part due to the microbes that are probably active in our systems. But this is also, because it's stuff coming from, it's your lungs, your airways, your mouth, your nasal system, so they're probably going to be bacterial components that create the uniqueness to individuals. But
they probably could do this. They probably could do this with the microbiome, but it wouldn't be a breath analyzer.
No.
Yeah.
You'd have to have a very specialized diet for that one too, I think.
You would. Right. Anyway, maybe one day, your smartphone with a little breathalyzer thing, maybe you instead of your thumbprint or a pupil scan to get into your bank app on your phone, you blow on your phone and your phone goes, "Oh, it's you. Here's all your money."
And it still won't work because we'll still be wearing masks.
Very possibly. Very possibly. And my final story for the night is a little bit itchy. You think about those face mites.
No, this is like yawning.
Why are you giving me the face mite story?
When you think about those face mites crawling on your skin, like maybe
I can feel them whenever you say these things.
Maybe they make you want to scratch. Maybe they start a little sensation of itch, and they make you want to scratch that itch. How does our body understand that physical itch sensation that can be caused by a loose strand of hair or a small parasite living on the surface of our skin? A study published in Nature this week has discovered that there is a subset of neurons that was previously thought only sensitive to chemical stimuli for inducing itch, but they discovered at the Scripps Research Institute that there are proteins, ion channels, for which the Nobel Prize was won recently. The piezo-1 and piezo-2 proteins, these ion channels, very important to sensations of touch. Anyway, they've discovered that these chemical stimulated itch sensors, these neurons in our skin, they also have these ion channels that allow to be stimulated by physical touch. And they never knew that before. So it's a researcher working in the Nobel Prize winner's lab, looking at the mechanisms of touches that lead to itching and scratching. It's really a misunderstood or understood area of sensory biology. And she put a bunch of these neurons in a dish and didn't know what was really going to come out, was staining them for the piezo ion channels as a control because they were not expected to be there. But then they found them. So the control ended up being the study. It ended up being something they did not expect whatsoever. And they found that these chemical stimuli responsive itch sensing neurons express the physical ion channels. And so it's a whole new pathway. And so they genetically modified mice to not have the piezo ion channel in their itch sensing neurons. And guess what? They didn't itch and scratch so much. And then when they gave the piezo one back to the mice, then they started itching and scratching more. And so this is a new understanding of these itch receptive neurons, that they have an ion channel that we didn't think they did before. So
did they cough as much? Because I was just thinking about how you get an itch in your throat, and it's a very similar sensation, but it has this kind of compulsion where then you have to cough.
Yeah. Well, they weren't looking
similar.
Yeah, they weren't looking at coughing in mice.
Yeah,
so
of course
I
not.
have no idea. But they were looking at this in skin. But since the epidermis, it's the inner lining of the airways is still kind of epidermal, it very well could be that physical. I mean, and the inner lining of the airway does have
next
little hair growth in it. Anyway, yes. We'll get rid of the cough. But while we understand how this touch-sensitive neurons, how these neurons are sensitive to touch and chemical stimuli, this could lead to additional ways to treat dermatitis or eczema or other physical itchings, things that get stimulated, disorders that lead to itching over time, itching and scratching significantly, which can really harm your epidermis, right? Can harm your skin, which is the barrier between you and the environment. So it's very important. And apparently 10% of people will suffer from chronic itch at some point in their lives. It's a lot of people who are just itching.
Yeah, you know who those people are? You know who those people are? Those are people who learn about face mites.
I mean, some of them, but not all of them.
Maybe you'll be scratching your face as you fall asleep tonight, Justin.
Yeah, I wonder why that is.
I'm going to say goodnight.
He's going to be smacking himself in the face.
One by one. Good night. Good night. Good night.
And I kiss you good night. And I kiss you good night. Good
and
night.
I'm
good
just
morning,
gonna
little face mite.
Don't mind me, you all sleep tight. I'm just gonna put myself under this UV lamp.
There was speaking about that, older men, not in their 50s, 40s, 50s, but in their 60s, older men in their 60s exposed to UV light while they were sleeping had worse sleep and more health problems than men than women or younger men. So there's something that happens as you get older, being exposed to UV light while you're sleeping, that's bad. So don't sleep with your UV lights on as you age.
Blue light is worse than any of them, and they can cause cancers and everything else because they mess with your melatonin levels or something in their something. I don't know.
Your face mites won't like that. You can't mess with your melatonin.
The best actual way to sleep is in darkness. Light therapy is a scam. Don't fall for it.
I love darkness. Hello, darkness, my old friend. Bringing face mites here again. Dun, dun, dun. It's time. We've hit the end of the show. I did it. We did it.
We did it.
We did it. We did it. We did it. Thank you so much, everyone, for joining us, bringing your face mites to the show. I hope they learned as much as we all did, and I hope that your little spaceship face, that it brought a little bit of entertainment to those face mites and to you. Thank you for joining us for sure. I want to say thank you also. Shout outs to Fada. Thank you so much for your help on show notes, show descriptions, and social media. Thank you, Identity4, for recording the show, Gord, Arnlor, others who help keep the chat rooms a nice place to be. And Rachel, thank you for editing the show. All of you. I couldn't do the show without you. Hosts, co-hosts, thank you. It's amazing. And thank you, TWIS patrons.
Yes.
Thank you to Teresa Smith, James Schofer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Gaurav Sharma, Ragan, Derek Schmidt, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollack, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Vote Beto for Texas, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Jackie Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, John Rodney Lewis, Paul, Philip Shane, Kurt Larson, Sue Oster, Jason Olds, Dave Neighbor, Eric Knapp, EO, Kevin Parachan, Aaron Lutzen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you for all of your support on Patreon.
A lovelier bunch of people there have never been.
Ah, what a lovely group of people. And you, if you're interested in being a part of that lovely group of people, you can find a link to support us on Patreon at twis.org. On next week's show,
we will be back Wednesday, 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and from twis.org/live.
Do you want to listen to us as a podcast? Maybe as you feverishly scrub your face before you go to bed? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to the stories and studies will be available on our website, www.twis.org. And you can also sign up for a newsletter that won't have anything to do with face mites.
I can't guarantee that. Yeah, we'll see. You can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into our junk inbox, and it will land as gracefully as a pumpkin toadlet. There.
Which is to say, not very gracefully at all.
No. It'll fall right off the screen.
You can hit us up for comments or story suggestions on our Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and complaints about the face mite story should all go to at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week in